Okay. So today the topic of our discussion is our presentation is encapsulation of the probiotics into milk-based functional foods. The speaker is Dr. Asutosh Mohapatra, who did his B.Tech in 2014 from our college, M.Tech in food processing engineering from National Institute of Food Technology, entrepreneurship and management from Tamil Nadu in 2016, PhD from Indian Institute of Technology, Delhi in 2024. So fresh graduate. Currently, he is working as a teaching-cum-research assistant at Management Development Institute, Gurgaon, since April 1, 2024. With that, I will turn over the discussion to Asutosh. And in the background, I'll share the link with people that are asking for it. Okay. All yours, Asutosh.
Okay. So good evening to everyone present over here. My respected faculties, dignitaries, my seniors and juniors. Thank you so much for giving the opportunity to present my PhD work in front of all of you. And I'm sure that without all of your support, my faculties, this work and this place right now, which I am here, it won't be possible. So just coming to the topic, this topic in the part of my PhD work, which I'll be presenting today, that is the encapsulation of probiotics into milk-based functional foods. This project work was carried out at IIT Delhi as a part of my dissertation work. So, as you all know, that probiotics are the living microorganisms. Just before that, let me ask if the slide is clear and it's visible to everyone. Hello.
It is, yes,
yes,
go
it's
ahead.
clear.
Okay.
Please go ahead.
Garage door.
Okay. So, as you all know about the probiotics, that probiotics are the living microorganisms. Either it may be a bacteria or yeast, which, when ingested in sufficient numbers, it gives multiple health benefits to our health. In a layman concept, we all consume probiotics in our daily life. Maybe it's a coconut water, maybe it's curd, maybe it's buttermilk, chhanch, maybe it's pokhal, which we are eating in the daily basis. It all contains probiotics in a sufficient amounts. As per the Food and Agricultural Organization and WHO, a recommended dose to get the sufficient health benefits from the probiotics, it was fixed around 1 lakh to 10 lakh CFU, that is colony forming unit, or amount of bacteria in a nutshell we can say. So when we are consuming 10 to the power 6 to 10 to the power 7 amount of bacteria daily, then we are supposed to get a sufficient health benefits from it. However, and when we talk about probiotics, the probiotics are a number of benefits to our human health, directly or indirectly. So just to reminder, during the COVID situation, most of the doctors prescribed lactic acid bacteria tablets. Along with all the antibiotics, they provide many probiotic capsules. It is because when we are consuming any kind of antibiotics, it kills both good bacteria as well as the harmful bacteria in our human gut and gastrointestinal tract. So when it kills the diseased bacteria, at the same time, the good bacteria are also getting dead. So to maintain or to increase the amount of bacteria or to balance those bacteria, we are getting additional amount of bacteria in terms of medicines. So similarly, when you talk about human gastrointestinal tract, probiotics are the first line of defense, and to adhere and to increase the immune system, and it always maintains a balance between the beneficial and harmful bacteria whatever present inside our gut. When the amount of good bacteria, that is probiotics, are getting reduced, we are getting different kind of diseases or disorders in terms of various symptoms. So it's like from the Ayurveda to till now, we know one fact: all the things started from our gut. When we have good food, good bacteria, then the whole body and whole physique of a human keeps good. So coming to the probiotic market, we know like till now, the milk-based products in global around constitute around 49%. of the probiotic market, followed by fruit and vegetable industries, followed by meat and bakery industries. So when you talk about the application of probiotics, a major part of application could be done on the milk-based products. In daily life or right now in the market, we are consuming Epigamia, we are consuming Yakult, we are consuming probiotic curd from different brands like Mother Dairy, Amul, Milk Moo. In probiotic ice creams are also there, probiotic desserts are also there. So in a nutshell, it is getting applied in various forms, most particularly into the milk-based products. So, and as per the statistics in 2023, the global revenue of probiotic market was around $134 million. However, it is expected to increase around $200 million by 2028. So, with this huge potential of market opportunity, we must say we need to, the market demands more kind of probiotic enriched dairy-based products as compared to other backgrounds like fruit and vegetable or meat-based products. So keeping those things, we thought of, okay, let's work on probiotic enriched dairy-based product, like to formulate those things. So when you think about any probiotic rich product, what's the issues with the probiotic market and probiotic foods? The major issues happen is most of the probiotic bacteria are getting where a food which contains the probiotic bacteria, those are getting died due to various factors like when it comes contact with the oxygen in the environment oxygen, if the moisture content of the food is increased or water activity is increased, then most of the then it is more prone towards the environmental barriers. If the product is not properly packaged, then again it won't be have good shelf life. What kind of storage temperature you are using to store the product, that also determines the shelf life as well as the probiotic shelf life of the product. And if the pH and acidity of the product is increased, and if the probiotic are directly getting contacted with the product, then most of the bacteria will get die, because at low pH and high acidity, any of the bacteria will get die. It doesn't matter it's a harmful bacteria or good bacteria. So keeping all these challenges, we think of a solution, like there are numerous solutions are there. So among those solutions, one of the important solution is encapsulation technique. Like what kind of, I'll go in details in the next slide, what kind of encapsulation technique we are using, what kind of wall material we are selecting for those particular encapsulation technique. And if we are using an encapsulation technique, then the output, whatever the product we are getting, what is the moisture content of that product? If it's the low moisture content, then we can increase the shelf life. Similarly, what kind of probiotic strains or probiotic bacteria we're using, because there are numerous bacteria of probiotics. There are heat sensitive bacteria, heat resistant bacteria, like mesophilic bacteria, thermophilic bacteria. So what kind of probiotic strain we are using, and based on the requirement of probiotic, like in the chat box, I 3 4 days before I saw one of our dignitaries asked about Sporlac tablet. So Sporlac tablet is a combination of Lactobacillus sporogens, and which mostly applied when we get any kind of diarrhea or what gut disorder. So to hinder the activity of antibiotics and to increase the amount of good bacteria, Sporlac is kind of a medium to increase the bacteria. And that bacteria particularly is Lactobacillus sporogens. Similarly, there are a number of good bacteria, Lactobacillus acidophilus, Lactobacillus serota, like in the Yakult we are consuming in daily life, that contains Lactobacillus serota. And another most important factor, whatever food you are consuming, whatever probiotic or encapsulated product you are using, it doesn't matter until unless it releases inside the human gastrointestinal tract. So if the bacteria, or we can see the core material, so basically encapsulation means one core material will be there surrounded by an wall material. So whatever core material is there, if it's getting out from the wall material and assimilated in the digestive system, that is the most important factor. Because if it is not getting released or not getting properly released or uniformly released, then there is no point of talking all these factors. So by, however, these are couple of solutions. If we can control these aspects, then we can have a good shelf life and good amount of bacteria in our human gut. So keeping these things, uh, like here I'm you can see like why we are talking about encapsulation. Encapsulation is basically a technology to encapsulate or to entrap any kind of solid, liquid, or gases in terms of a capsule or particles. Normally, basically, it gives some protection to the particular target material. For example, if I am taking liposome, then I am using a certain kind of wall material. For example, say alginate. So here alginate will be the wall material. Liposome is our main core material. So alginate must give some kind of good protection to the liposome. At the same time, alginate should maintain the proper release of the liposome at the target site. And also the overall aspect, whatever wall material we are using, it should give a proper acceptability, because if the taste won't be good or the product won't look good, normally we won't buy that. And also uh, using encapsulation, we can have a better storage, and we can try and we can formulate different functional foods. It may be a nutraceutical, it may be a vitamin enriched food, it may be any kind of mineral enriched food, and depending upon what kind of particle size we want. It may be a nano encapsulation, like in the nano range, if we want the size of the particle, final particle. If we want the micro size of the particle, that is micro encapsulation. There are a number of techniques are there. Like here, I have mentioned couple of techniques based on the particle size requirement. It may be electrospraying, spray drying, extrusion, freeze drying, the combination of spray and freeze drying. There are numerous number of techniques are there. However, among all these techniques, freeze drying is one of the best techniques in terms of maintaining the quality. When you talk about any probiotic bacteria or making the strains of probiotic bacteria to keep for a longer time, we usually adopt freeze drying technique because it maintains the quality as such, and it didn't bother any kind of heat treatment, so there is no chance of any bacterial death. However, again, it takes like very high, uh, what you can say, like the electricity consumption. It is comparatively 10 times more than any kind of spray drying. And the major problem is the time. Time requirement is around 8 to 12 hours or 13 hours. That hinders the use of spray freeze drying in the industries. That's why most of the industries go for spray drying technique, where they can easily get the powder uh, within a short period of time with somehow good amount of encapsulation efficiency. So whenever we talk about any kind of encapsulation technique, the selection of wall material are the most important criteria. So the wall material must give, there are some selection basis are there. The wall material must give some kind of functionality to the product. It should be food grade material. It should have a good barrier properties from the environment, oxygen, and from the other adverse conditions. It must give maximum protection to the core, so that, sorry, the core material should be reached to the targeted site. And whenever you are using any kind of wall material, you are selecting, the wall material must have good rheological properties, so that it would have good flow properties and it could withstand the process of transportation and storage. And at the most, it should be generally recognized as safe as per the FDA standards. So based on this criteria, we need to select our wall material. Accordingly, we need to select our encapsulation technique based on our type of requirement of particle size and what kind of material we can use. So with all these things, we come up with some kind of research gaps. That is, the first is the poor survival rate of any probiotic bacteria due to low pH and high acidity inside gastrointestinal tract. Why it's there? Because when you talk about human GIT, the normally any of the food you are consuming, it will first go to the stomach, subjected to the lower intestine, and then upper intestine, large intestine. So at the stomach, the pH is around 1.5 to 3.5. Any of the bacteria which are open and alive will be immediately died at that low pH condition. And similarly, in the stomach and in the intestine, the presence of bile salt the various pH and the immediate shifting of pH that creates a main barrier and main issues which reduces the viability of any bacteria. It, in those conditions, it doesn't matter how much bacteria we are consuming. For example, we are consuming 100 amount of bacteria. When it reaches to the large intestine, maybe 10 or 20 bacteria are getting alive. So in this context, we need to understand that how we need to increase the survival rate of the bacteria. As similar, whatever we are consuming, the same amount should be delivered at the intestine. Also, there are lack of different strategies to improve the technological and gastronomical robustness. Uh, the non-uniformity of the control release inside the GI tract, whatever bacteria or probiotic or bioactive compound we are consuming. The release kinetics and release uniformity should be there, which is not usually present in most of the cases. Uh, Also, limitations with different conventional encapsulation techniques with lower encapsulation efficiency and recovery yield. So, keeping all these points, we concluded or we formulated a couple of objectives for as a part of my research. The first objective was to optimize the wall material using to spray encapsulate Lactobacillus acidophilus. Here in our subject, we took Lactobacillus acidophilus is a thermophilic bacteria as well as a good probiotic bacteria, which increase different or which confers different health benefits to our health. So, and here we choose spray drying as it is one of the widely accepted encapsulation and drying technique in the industrial scale. So, uh, the first was to optimize the wall material combinations to encapsulate the Lactobacillus acidophilus. Second objective was to study the physiochemical and biochemical characteristics of the encapsulated probiotic powder and to understand the control release kinetics inside the human gastrointestinal tract. And third one is to incorporate that encapsulated probiotic powder to formulate probiotic enriched Indian curd. As curd we are daily consuming in daily basis, so we thought of okay, let's make a probiotic curd and to incorporate that encapsulated powder, and we will further study its physiochemical, biochemical, rheological, and sensorial attributes. So overall, this research demonstrates we tried to develop or try to understand the feasibility of developing spray encapsulated probiotic powder with enhanced bioavailability and sustained release inside human GIT. And second was to successful formulation of probiotic enriched functional curd uh, to use it or to eat it in our daily dietary consumption to strengthen the gut microbiome. Coming to the first objective, that is the optimization of wall material, we here selected three kinds of wall material and their combinations. Uh, the first one is the skim milk powder, second one is the maltodextrin, and third one is the cellulose acetate phthalate. All three are widely skim milk powder and maltodextrin are widely accepted wall material in terms of food supply chain due to their various good properties. And when you talk about any kind of fruit juices, any kind of spray dried product, maltodextrin is kind of a general material to reduce the hygroscopicity, because when the sugar content of any liquid or any fruit juice is high, at that time it will stick around the surface of the whole spray dry. So that time if you are using or adding maltodextrin at a certain amount, then it will reduce the stickiness and hygroscopicity of the product and we can get a good recovery of the final product. So these two are the selection of spray dried for the spreading things. However, cellulose acetate phthalate, that is a biopolymer which is mostly used in the pharmaceutical industries. Whenever you are taking any kind of capsule or medicine, the outer layer is mostly composed of cellulose acetate phthalate. Why? Because it is first the inert and inactive in nature. It doesn't react with the core material, and it will only dissolve the if we are using as a wall material, it will only dissolve when the pH will be greater than 5.6 or around 6. If the pH will be greater than 6, then only the cellulose acetate phthalate will only dissolve. So when we talk about human gastrointestinal tract, at the stomach, the pH will be very lower, around 3.5, 1.5 to 3.5. That time it will give some kind of protection to the core. When the at the large intestine, when the pH will be greater than 6, at that time this wall material or this particular biopolymer will dissolve in the intestinal juice, and the core material will get released. So therefore, we thought of, okay, let's try these combinations of these three and see how how much encapsulation efficiency we are getting for the particular lactobacillus acidophilus. Therefore, we to optimize the combination of this cellulose ester phthalate, maltodextrin, and skim milk powder, we tried a particular design, central composite rotatable design. But however, in the spray drying, we fixed some kind of parameters based on the preliminary trials, that is inlet temperature, feed flow rate, aspirator flow rate, what of the nozzle diameter will be there, and atomization pressure will be there. This kind of variables we fixed based on the preliminary trials. The independent variables we took three, that is the wall material different combinations, and the response variable we studied, that is the survival rate of the bacteria, what are the encapsulation efficiency of the bacteria we are getting, what is the moisture content of the final powder, and what is the ultimately recovery yield of the powder we are getting. And like this, you can see this is a schematic representation. Here we took the emulsion that is that is comprised of skim milk powder, maltodextrin, and cellulose phthalate with the active culture of Lactobacillus acidophilus. Using a feed pump, the whole emulsion was subjected to the main drying chamber through an atomizer and through a high pressure nozzle. The nozzle diameter we can vary. However, here we took 1.5 mm. Based on our requirements, we can change the nozzle diameter. When the hot air and the emulsion liquid are getting contacted, it is a co-current system. So hot air and the emulsion are liquid are in the same direction. So when the contact of hot air and emulsion happened, at that time most of the liquid particles are getting dried, and whatever the remaining particles are there, through the nozzle, it will fall down in the main chamber, that is the product collector. So most of the heavy particles will fall down, the dried heavy particles will fall down, and some of the lighter particles will be segregated to the another production collector through a cyclone separator. So this is the overall, not like representation of any kind of spray dryer. We have a cyclone separator to separate the lighter particles from the heavier particles. And after the drying happened, we need to collect the sample from all the parts. It's the main drying chamber, product collector, and from every chamber, we need to collect we can collect the powder. So therefore, the recovery will be much more higher as compared to other drying techniques or other encapsulation techniques. But the prediction of the response variables, that is the survival rate, encapsulation efficiency, moisture, and yield, there are a number of prediction techniques are there, or optimization techniques are there already we are using. For example, RSM is there, response surface methodology, and there are various techniques are there. However, here we tried to combine artificial neural network with genetic algorithm. So why we did this? Because a combination of ANN and genetic algorithm will increase the efficiency of the overall independent variable system. That is, when you talk about the combination of skim milk powder, maltodextrin, and cellulose ester phthalate, it will try different kind of iterations, training and testing, and after that it will increase the efficiency of the overall expression. So for the prediction, we use artificial neural network. For the optimization, we use genetic algorithm, in which we try to maximize the survival rate, encapsulation efficiency, and recovery yield. And these are the major targets which need to be enhanced, which needs to be maximized to get the good goal. And the log reduction and moisture content should be minimized. So keeping this criteria and feeding the whole coding system, we got the optimum value of our wall material concentrations. That is, skim milk powder should be around 10%, maltodextrin should be around 11.28%, and cellulose ester phthalate will be around 2%. However, these are subjected to a particular TS, total solid of the product. Here we try to keep the total solid around 30%. So from the 30%, this will be the combination. So right hand side, as you can see, these are the spray dried probiotic Lactobacillus acidophilus powder we finally could develop using the spray drying technique. So now we got the powder, spray-dried powder, probiotic powder. So in the second study objective, we studied the physiochemical and biochemical characteristics, in which though after studying all the physiochemical characterization, we understood that the spray drying enhanced the powder characteristics and flow properties of the particular spreader powder. We had a good bulk density, reduced flow properties, behavior, reduced hygroscopicity, and the moisture content was around 6%. When normally we recommend a moisture content of 4 to 7% for any kind of probiotic spreader powder, because there need to be some kind of moisture. Otherwise, whatever the bacteria present inside the uh, powder, whatever the bacteria are there, it won't get moisture to carry out the normal functions. So there should be some kind of moisture content. However, the recommended was 4 to 6 or 7%. In our case, we got around 6%. The color of the powder was white and it's good. The stability of the emulsion was good. The particle size analysis, here you can say bimodal, here I wrote. It means the more kind of there are mono layer, bilayer, tri-layer, different kind of particle size distributions are there. More span value will be there and bimodal will be there, then it gives to a good shelf life, good storage conditions. The same analysis, the scaling electron microscopy, we can see the particles are mostly spherical in nature. So when we talk about spherical in nature, also at the same time we conducted the biochemical characteristics of probiotic powder, which include, we tried to develop the simulated conditions of our gastric juice, intestinal juice, and different pH. So why to the bacteria, whatever we developed, it must survive the harsh conditions like the human gastrointestinal tract. So when we try to simulate those conditions of human GIT, and we subjected the probiotic powder to those different fluids, and we studied whatever the survival rate of the bacteria at the end of the digestion. We assumed the digestion of whole human digestion is 4 hours. So accordingly, we calculated the survival rate of the bacteria at the end of the digestions using the simulated gastric fluids and simulated intestinal fluids. So this is some preparations of the basic techniques for the gastrointestinal digestion. So from this, we found out that in the simulated gastric and intestinal conditions of 4-hour duration, the encapsulated powder has achieved a survival rate of more than 6 log CFU. 6 log means 10 to the power 6 CFU per gram. However, the control sample, that is free bacteria, was around 2.3 to 2.4 log CFU. And in the gastric and intestinal juice, simulated juice, from the encapsulated powder, the bacteria of around 26% got released. Release means got died. However, in the control sample, it was around 76%. So you can see the huge difference, that the wall material was efficient enough to maintain the bacterial viability, the Lactobacillus acidophilus viability inside the released inside the gastrointestinal juices. When we try to subject the bacteria at different low pH conditions, that is 2, 3, and 4, in all the cases of the exposure to low pH conditions, the encapsulated powder showed good viability of more than 7 log CFU, which is the required target of FAO and WHO that we could achieve as compared to the free bacteria. After that, we tried to study like if we are getting consumed that particular probiotic bacteria, how it's getting released inside the gastrointestinal conditions. So as you see here in the it's the morphological characterization showed that the bacteria are spherical in nature. So for spherical and heterogeneous sample, heterogeneous in the sense, here we have different kind of wall matrix and a core material. It's not a single wall material. So for a spherical heterogeneous sample, there are already established release kinetic models in the pharmaceutical, like for most of the pharmaceutical industries, they go for this studies, behavioral kinetic studies. So we selected these 6 models: zero order, first order, Hickson Crow, Higuchi, Kurzmer, Pepas, and Bethel-Nusselt model. And we try to understand the release mechanism, how it's getting released. It's kind of a predictive modeling. And ultimately, at the end, we found out that for when the product is getting released inside the gastric fluid, simulated fluid, and pH 4, it follows zero order model. When it's getting released inside pH 2 and 3, it follows Kurzmer-Pepas model. And the release mechanism, we found out 2 types of release mechanism here. I'll explain. That is anomalous transport, another is super case 2 transport. So what are these particular transport? There are a number of release mechanisms, we can say release transport mechanism inside the human GIT, like Ficinian transport, anomalous transport, case 2 transport, super case 2 transport. All this transport mechanism has some kind of different mechanism of release. In our case, we understood that anomalous transport, which is a time dependent transport, which is mostly governed by the erosion based release and diffusion based release. Whatever the core material or bacteria are there inside the capsule, encapsulated capsule, due to surface erosion of each layer and due to the diffusion process, the inside core material is getting out from the wall material. Another transport mechanism was we found in our case, that is super case 2 type transport, that is a time independent transport. Here, the main governing mechanisms are erosion-based mechanisms, swelling-based mechanism, and what are the polymeric chains of biometal are there, those are getting relaxed. And due to the different adverse stresses, the outside of the encapsulated particle, due to those stresses, whatever inside core material are there, those are getting out of the encapsulate. So this particular transport mechanism we could understand in our case. However, it will be varied if the size of or shape of the particle will be different. For example, it's a I can say hexagonal shape or a cylindrical shape. That is a cylindrical capsule we kind of eating in the daily life. But all those different various shapes and size of the capsule, the mechanism of release and transport will be different. When we try to store, when you study the storage study of the encapsulated powder, we could clearly at 2 different temperatures. One is refrigerated temperature, one is ambient temperature. We finally could understand that the Lactobacillus acidophilus in both the cases, the amount was more than 6 log CFU after the 6 months of storage. If we are keeping in a ambient condition, up to 6 months, it can maintain the viability of bacteria more than 6 log, which is a very much desired properties and it can say it's a very good validation for the encapsulation technique we have adopted. At the same time, we try to conduct accelerated shelf-life study for this particular encapsulated powder at different temperature combinations, and we determine the specific rate constant value, that is K value, what is the inactivation rate constant. This study can be mostly useful when you are going for long-term storage preservation system in the industrial scale. That time, using this specific rate inactivation constant, we can give some kind of insights for a long storage system. So just coming to the last third objective, that is incorporation of encapsulated powder to formulate Indian curd and study of its physiochemical, biochemical, geological, and sensorial attributes. Here, what we tried to do, we formulated the standardized curd. After making the standardized curd, we added the encapsulated powder at different levels. That is 5%, 7.5%, and 10%. You can ask why we didn't go beyond that. There are a couple of studies, like for example, in the Kharagpur, ID Kharagpur, H.N. Mishra sir, he has conducted a couple of studies for this probiotic powder, and also some kind of studies in the abroad, which are saying when you are going to add the probiotic powder additionally in the any kind of liquid or semi-liquid product, we shouldn't go beyond that 10%, as it might change the whole properties of the liquid solution. So that's how we kept it up to 10%. And after that, we started the physicochemical, textural, biochemical, rheological, and sensory study for all the curd formulations. In a After adding at different proportions, we understood that the addition of encapsulated powder in the curd enhanced the physiochemical and textural properties of the curd. Like it had a good water holding capacity. Whenever we are seeing a curd, the synersis should be less. In our case, the synersis was very much reduced, significantly reduced. Water holding capacity was increased. The pH was slightly changed. Didn't much changed after addition of the powder. The firmness and cohesiveness of the overall curd was very good. The biochemical characterization, that is, the curd was subjected to the simulated gastrointestinal fluid, gastric fluid, simulated intestinal fluid, and different concentration of bile salt. Because at the same time, we already studied for the powder. But if you are proposing the probiotic curd must contain the sufficient amount of bacteria when it reaches to the large intestine. In our case, we understood we found that after with increase in the addition of probiotic powder, the survival content of the overall lactic acid bacteria was gradually increased, and in all the cases, the viability was maintained more than 8.5 log CF. In the market, whatever probiotic curd we are having, Amul and Mother Dairy, uh, those are maintaining a level that 10 to the power 6 bacteria are added are there at the time of consumption. However, they're not mentioning what are the amount of remaining bacteria are there at the time of digestion. In our case, we found out that at the time of addition, the bacterial amount was around 10 to the 12, and at the time of after the digestion, the survival rate of the bacteria is around more than 8.5 log CFU, which has multifold increased the amount of probiotic bacteria in the human GIT. When you release the bacteria in different bile salt concentration, all the wall materials could protect the bacteria efficiently and increase the cell viability inside bile salt concentration, and the overall release were also very less for control sample, that is plain curd. The release of the or death of the lactic acid bacteria around 55%. In after with increase in the addition of the powder, the death and release of the bacteria was very much reduced to 10%. Rheological properties of the probiotic curd also showed, like when we were increasing the when we were adding the powder content, the overall TS of the curd is gradually increased. So whatever amount of powder you are adding, it doesn't matter, but the curd should be looked like a curd, and the addition of the powder must not change the rheological properties of the curd significantly. In our case, we found out that after the addition of the curd also, after the addition of probiotic powder also, the curd is behaving like a soft or weak gel, which is the standard one, and the gelling strength of the curd was gradually increased, which is a very good finding in this study. At the end, we tried to conduct a sensory analysis using 5-point hedonic scale with trained and semi-trained panelists, and we tried to model the sensory attributes, that is taste, flavor, texture, and color of the curd. After conducting the physiologic modeling, we understood that the best curd sample as per the panelist was sample 4, that is control sample, that is obvious. Followed by the 7.5% added curd, powder added curd, was mostly accepted by the many of the panel members, followed by 5% and followed by the 10%. So in overall conclusion, like we found out that we tried to model and predict the wall metal concentration using artificial neural network and genetic algorithm. At the same time, we also compared with the RSM technology. Like we also did those things. As compared to that, it efficiently predicted, validated, and optimized the wall metal concentration. We could achieve an enhanced bioavailability and stress adaptation of the encapsulated powder, and the cell viability was more than 7 log CFU during the release inside the simulated gastrointestinal conditions. And two types of release mechanism we could understand that governs in our case. The encapsulated powder exhibited improved physiochemical and thermophysical properties. The addition of the powder to the curd showed better retention of lactobacillus, of the lactic acid bacteria more than 8 log CFU. And the storage study of the powder showed that the cell viability was maintaining up to 6 months with more than 6 log CFU, which is a very good in terms of ambient storage conditions. In the refrigerated storage conditions, it was around 9 log CFU. In the ambient condition, it was more than 6 log CFU, up to 6 months. So from this study, we could understand that, okay, there are some kind of gaps are there which also could be addressed in the future. Like when you talk about different combination of wall matrix, we should try also different prebiotic and probiotic combinations. Already some kind of literatures are there. So prebiotic are the food for the probiotic. So when we are using some kind of prebiotic, for example, casein, if we are using or any carbohydrate we are using as a wall metal, at the same time it will be used as the food for probiotics. So it will add more kind of multifold increase of survival inside the human GIT. Those combinations we can try. And when you are talking about spray drying, we need to we can add multi-step optimization techniques. And here we understood for the spherical shape of morphological size, we calculated the release kinetics. If you are making different shapes and size, for example, octagonal, hexagon, cylindrical shape, then in that context, what kind of release behavior and mechanism it will follow inside the human GIT, that we can understand. And accordingly, we can engineer those kind of shapes depending upon what kind of probiotic functional food we are trying to formulate. So for this research, like I am truly acknowledging the lab, food customization research lab at IIT Delhi, the facilities we used at Central Research Facility and Nano Research Facility, NDRI kernel for providing the culture and some kind of suggestions at the time of requirement, the Neptune Sony for outsourcing couple of experiments, ICT Bhubaneswar for conducting the moisture substance analysis. So because without them and without all of your faculty's guidance, this work won't be possible. So with this, I'm thanking all of you for listening to the present. I hope I increased or I took a little bit more time, but at the same time, I am happy that I could present in front of all of you. So thank you so much, sir. Thank you, everyone.
No, thank you, Asutosh. Excellent presentation. Many good discussions came out of it. So now we'll open it for questions. I see that some of you are joining right now, but those who are already here could start with your questions. I see Biswajit is already ready.
No, I'm not actually ready. I'm waiting for Dorau to start. I just unmuted myself. I thought Dorau should be given the first chance to start this. Anyway, Asutosh, I noticed that in one of your slides that if encapsulated bacteria, I will call it encapsulated bacteria, if it is subject to this thing GIT juices, then there's a survival rate of 26%. But if it is in a normal case, it will be about, destruction rate of 26%. And in a normal case, it will be 76%. 76%.
Yes, sir.
Yeah, that's correct, right? So which means that your the encapsulated bacteria will have a survival rate of almost 3 times that of normal bacteria, right? Now, the FAO standards of 10.7 or 10.6, 10 to the power 6 or 10 to the power 7 is, I am assuming that it is the intake that a human being is supposed to take, and whatever is the destruction that happens, the balance has to reach to the GI, to the gut.
Great, sir.
Yeah, so in your case, which means in encapsulated bacteria, we can use 1/3 of that. Am I correct in that?
Yes, sir. Right.
Okay, so which means the total population of the culture that we will have to use will be less, is it?
Yes, exactly. Yes, sir.
Okay. So instead of 10.7 or 10.6, it will be one third of that, whatever is the calculation. Right?
Yes, whatever
Okay.
sample initial concentration of bacteria we are taking, we can
Yeah.
reduce that content, and the remaining two-third we can use for the next batch. And at the
Yeah, of
same
course.
time,
Yeah.
yes,
So
sir.
that which means the cost of production reduces.
Exactly, sir. Exactly.
Right.
Yes, sir.
Yeah. Okay. So that was one clarity I needed. The second one was that you mentioned that you produced curd from the encapsulated bacteria, or was it
No,
that the
sir.
curd was
No,
made and
sir.
this was added?
So, yes, sir. The curd was normally made using the standard protocol. After the formation of curd, we added the probiotic powder and just mixed it uniformly. So the sir ultimate,
Okay.
I'll add one thing. The ultimate thing is to deliver a sachet or which contains just like Maggie inside the Maggie masala are there. 1 rupee Maggie masala. Similarly, one sachet will be there that contains the probiotic powder. Whenever we want to eat uh, any kind of probiotic supplements, we'll just here the sachet will mix in any kind of, for example, say buttermilk, curd, yogurt, ice cream. It doesn't whatever the uh, main
Okay.
sample will
Understood.
be there. We'll just mix it and we'll drink it or we'll eat it. In that context, it will give it like very kind of potable, easily and like very easy to use at the time of consumption. No need to use very kind of different hard technique. You will just tear it, mix it, and we'll drink it. That's the whole concept we are proposing for the market.
Okay, and which is why you have tried to store it at normal room temperature, and your
Yes, sir. Yes, sir.
experiment was
Right.
successful, right?
Exactly.
Okay, so
So
which
for up
means
to 6 months,
you are easing the delivery system of the entire thing. That is the whole purpose of this.
yes, sir. So we tested like up to 8 months we tried for the storage conditions. So up to 6 months at the ambient condition, it was maintaining more than 6 months. For the refrigerated condition, it was maintaining more than 8.5. That's okay. But for the ambient condition, after 6 months, it is maintaining the liquid level. After 6 months, the bacterial viability is getting reduced.
The potency
And
goes.
we tried the packaging material here, the glass bottles. However, also another study can be done if we are changing the packaging material. Then what are the like how the death kinetics of the bacteria are getting changed? That also we need to study. But again, it will vary from different packaging material to packaging material. In our case, we used simple glass bottle, small glass
So
bottle.
that will be your next next trial or next experiment
In the
in
future.
India, right?
Yes.
Okay. Yeah, all the best, and
Thank
all
you.
the best for your patent also.
Thank you so much, sir. Thank you.
Okay. Dora babu, ଆପଣ କଣ ପଚାରିବେ କି?
Actually, it's a very interesting topic, and because he has already done the research and research study, it's welcome, and he has found a lot many things. Actually, I was wondering only one thing, that in case of drying, normal drying of milks as a milk product, we are going for two-stage drying. First, we are concentrating to a stage for a certain moisture content, say from 13% to say 87% to it is brought down to 45%, and again we are going for spray drying. But here, whatever presentation I saw, it is directly being taken for spray drying, which have either a temperature shock in a differential temperature. The temperature range is very high, and second, there is a moisture content difference is there. I was wondering, in case I go for the two-stage drying system, one first evaporation and second for spray drying, will it have any effect on its content, final content? That's what, or is it not possible?
Yes, sir. To
That's
answer
what
your question, definitely it will change the ultimate powder characteristics. However, to go for a two-stage system, it has some purpose. Why we are going for two-stage? For dairy waste conditions or milk, there is certain purpose. In our case, we don't require that purpose. At the same time, we are reducing the time interval. If we are going for two-stage, then it will increase the time of whole operation. Here,
That's right.
using just single stage drying,
that we can go for multi-stage drying also. But in the single-stage drying, we are getting the sufficient number of bacteria in the powder, and the drying, using drying, we are reducing the moisture content to a recommended level. We could achieve those facts. So if you already achieved those particular targets, then there is no need to go for multi-stage drying for this particular bacteria using the wall material combination. And of course, you are right, sir. At high temperature, here we're using around 150 degrees Celsius. At high temperature, any bacteria will get die. But that is the whole point of encapsulation. We tried different combination of wall metal, which has individual characteristics, which will impart those benefits on the core material to protect at high temperatures. So, and at the end time, to be honest, we tried at different temperatures also and different flow rate combinations. Because if the flow rate is less, the temperature is high, then there is more chance of bacteria getting died. So we tried the combination of different flow rate, different temperatures, and we optimized the combination was 150 degrees Celsius and 2 ml per minute. Using those combinations, we optimized the combination of wall material. And once the wall material optimum conditions we achieved, then we found out the probiotic powder at the optimum condition. So in the nutshell, we reduced the time interval. We could achieve the sufficient amount of bacteria and with a good shelf life.
But is it the research has been limited only to the one product cod, or have you gone for any other extra product, dairy products or milk products?
For this, we tried, officially we tried for curd. We also went for buttermilk, like just for side work. In the buttermilk also, we are getting the solubility of the powder was reduced. Why? Means the basic concept was to add it immediately and drink it in the buttermilk. But when we'll mix it in the buttermilk, the problem is happening that it is not mixing uniformly. First thing. Second thing, the product is not the powder is not getting soluble, which also another good, because why? If the product will be powder will be soluble immediately, then there is a chance that the core material will getting out of the wall materials. And
I can suggest a solution for that. Asutosh, I will talk to you separately. But Dorabu, he had mentioned that the total solids of the solution he was using is 30%. So that could be one reason why he went for one stage only. But yes, it can be done at 2 stages. So.
Okay, let us go for other. Okay. Let us hear from others.
Ramo, you had a question?
No. Hello?
Yeah, we can listen. We can hear you.
Actually, I joined late. So actually, experts are there. One concern, only 12 people are there. So that has to be discussed.
Sunagal, can you repeat, please?
He is probably saying only 12 participants were there. That is the problem.
Okay. Yeah. We should increase that. Well, anyway, this is reaching out to a lot of people through the YouTube channel. So we are good. I have
some question.
Yes, go ahead.
Yes.
Go ahead.
Hi, Asutosh. This is Sabyasachi.
Namaskar. Yes.
Namaskar. It's a good work. I have some simple questions.
Yes.
First question is that at the present condition, what the study says, there is a lot of report around lactose intolerance.
Yes, sir.
Right. So in that condition, how do you balance out the health benefits of probiotic with the lactose intolerance that we are having with the consumer?
Certain kind of probiotic bacteria are there which could reduce the lactose intolerance. For example, Lactobacillus sporangiens, Lactobacillus delbrueckii. So this kind of particular bacteria has the capacity to reduce the lactose intolerance. If you are using those kind of bacteria in the daily conditions, either by encapsulated form or diet form, whatever form, or for example, yogurt has rich in delbrueckii. So if you are getting yogurt at the daily conditions, then there is a much more better chance that we can reduce the lactose intolerance in the general basis.
Yeah, the second question that I have is, yogurt itself is a natural probiotic.
Yes, sir.
So how do you justify adding some additional probiotic to that?
Yeah. So, sir, many of the dairy products are naturally probiotic in nature. here we are not just maintaining probiotic, we are increasing the number of, we are increasing the survival rate of the probiotic. Whatever technique we are using, that's a separate thing. Just say if the yogurt already has a limit, particular 10 to the power 7 amount of bacteria, we are trying to propose if the bacterial amount is getting increased at the time of digestion, then we will get more benefit because at the market already curd are there. At the same time, we are in the market, we are also getting probiotic curd. So why this requirement happened? Because when you are using the normal curd at the after the digestion, whatever the recommended amount of bacteria are there that are not getting alive. So to balance that dead bacteria, we are additionally incorporating extra bacteria to maintain that balance, and we are talking it a probiotic. In our case, we try to propose that okay, at the time of digestion itself, we will increase the bacterial amount to a very high amount, so that we need at the initial stage of consumption, for example, we need a 10 to the 10 bacteria, then only, and we are checking at the time of digestion, 2 logs if we are decreasing. So we're proposing, okay, after the digestion, 10 to the 8 amount of bacteria getting alive. But at the market samples are there, we try to study for mother dairy, probiotic curd, and also this Yakult also. They're also mentioning that at the time of consumption, this amount of bacteria are there. But to be honest, practically, that is not maintaining in actually. And at the time of digestion, that amount was getting very much reduced. So to cope up that thing, we proposed, okay, at the time of digestion, initially consumption, obviously the bacteria should be higher, so that it will maintain a sufficient balance at the time of digestion. That is the whole motto for this work, sir.
So did you go for in vitro digestibility test?
Yes, sir. Yes, sir. Yes, sir. Simulated conditions of intestinal fluid and gastric fluid.
So
and
how much was the loss?
Sir, for the encapsulated powder, the loss was around 26% at the time of complete digestion, and for the free bacteria, the loss was around 76%. Around 3 times improved.
So that 26% from where to where?
Sir, from the time of addition to the end of digestion, like we understood that digestion is a 4-hour process. So we first made the intestinal gastric fluid, in that we subjected from 0th time, like 0th hour, we subjected the powder inside that. Then we went for simulated digestion condition. Then at the end of 4-hour digestion, in the intestinal fluid, we checked what is the survival rate of the bacteria. So initially we had 10 to the 12 bacteria. At the end of digestion, we could finally achieve around 8.5 log bacteria. So for encapsulated powder, around 3.5 log reduction was there. However, for the free Lactobacillus acidophilus, the reduction was around, initially it was 10 to the 12, and at the end of digestion, it was around 3.2 logs, 3.2 logs CFU. So a significant reduction for the free bacteria, but a very less reduction for the encapsulated powder throughout the gastric digestion.
Okay, that was a nice presentation and answering of questions. Good luck.
Thank you, sir. Thanks, sir.
Let me follow up with just one last one from follow-up of Professor Sabyasachi Mishra's concern. Okay, so in South India, mostly people eat hot food, and then they eat curd after that. Okay.
Yes.
And that's done for a purpose. Okay, so here you are talking about people taking antibiotics and then killing all the good bacteria or whatever profitable bacteria. Then we add probiotic as another capsule into it. So the culprit is actually what? The antibiotic.
Drugs.
Huh?
Mostly,
Drugs.
yes.
Yeah. The antibiotic is the big culprit here. Okay.
Yes.
So for good health conditions, in good health conditions, one can just survive without, like 50 years back, antibiotics were very rare, and people used to survive with this kind of. So can we add chili powder and probiotic? I'm bringing up a chili powder into the capsule and say, hey, now we have a I don't know, bacteria killing, bacteria surviving mechanism. This is a very jocular question, but I want you to think about it.
Sir, all these masalas actually act as preservatives.
Add what?
Acts as preservative. They kill the bacteria, both good and bad together.
Yes.
The masalas. Acha. Okay.
Yeah.
Masalas, oils, there are good preservatives, sir. But sir, ultimately, antibiotics we need, because in the present condition, whatever the environmental conditions are there, whatever the air we are intaking, obviously we are getting a lot of disorders. As before 15 years back, we are not getting that much diseases as compared to right now. So in some way, we already reduced or weakened our immune system. So for that, we had to take antibiotics. But to compensate the negative aspect of antibiotics, we need some good supplement. Like mostly doctors used to prescribe, if you are getting a fever or any kind of gut disease, you take coconut water daily. That's the basic concept from our elder generations also. Coconut water, coconut water has very good probiotic abundance. When you take coconut water, so basically in the empty stomach, it will immediately enhance the good viability of the bacteria, and it will increase the probiotic conditions. So coconut water, when you're taking any kind of hot and spicy foods, okay, let's take curd, because at the same time, when spiced food are killing the any kind of bacteria, good or bad, curd will compensate those good bacteria and maintain a balance in the our intestine and stomach. So to maintain that thing, or we are getting diarrhea, or for example any kind of loose motions, let's take buttermilk or chach. So all these will indirectly increase the good bacteria, and once the good bacteria in our gut will increase, all the problems will slowly resolve indirectly and directly, because ultimately it will improve the immune system. Once the immune system will get improved, automatically all the things will get resolved. So all the problems started from the gut. Once we are having a good gut, we are having a good digestion, many of the problems will get resolved.
No, to add to that, Asutosh, actually what has happened, our Indian, food habit that we have been following for so many years, centuries, we are just going back to those things only.
Right.
Yes, sir. Yes,
all these
correct.
food supplements that we are talking about, it's the innovation from US, sir. I'm returning back to USA from India. So there, the food habits have changed. They do not take I am sure, sir will agree with that. Antibiotics is strictly, very restricted in US conditions. So, but because of their food habit, they are lacking all these health benefits of different foods.
Yes.
And all these food supplements that has come, most of them are innovated in US.
True.
But Indian condition, as rightly said, we have all these natural and normal food practices and that we have in South India or whatever, even in Odisha, pakhala.
Yes,
That is I think that is the best probiotic that we can have cheaply.
Absolutely, absolutely. Okay, so it's 8:36 in India. Thank you very much, Asutosh. We can continue with this discussion in CIT and forum. It was an excellent presentation, and all the best with your future.
Devo, I think Dr. Sudhanshu Mananda has been waiting for some time.
Do you want a question, Sudhanshu?
No, sir. It is a nice speech. Actually, my network is not working.
Oh, okay.
No issue.
All right. So we can continue with the discussion in the forum, and you should be more active in the forum.
Yes,
okay.
Yes,
That way we can interact with you and learn from you a little bit more. So this was a learning experience for me, at least. But I know that experts are there around you, and they might guide you better. But I will be the learner in the group. So anyway, thanks a lot for sparing the time, and hopefully we'll see your participation in the future in the tea talks. Okay. Have a wonderful day, all of you. Rest of the evening.
Thank you so much.
Thank you, sir. Thank you.
Thank you.